Bacteria are little factories for all sorts of materials we use in real life, from insulin to proteins for medical research. But these little microalchemists are teaming up across species to make even more sophisticated real-life materials, including some that we might wear. We've previously covered bacteria that were able to produce faux leather. Strictly speaking, bacterial cellulose. While plants are usually the organisms we think of as the big cellulose producers, bacteria can do it too, such as Comagotebacter salinus, the bacteria that synthesizes kombucha. So, when producing synthetic textiles, researchers turned to K. salinus to make this cellulose-based material. Bacterially derived cellulose textiles would be a tremendous W for environmental friendliness, especially since so much of our clothing is made from petroleum-based synthetics. Cotton, linen, and wool may come to mind with clothing, but about 60 to 70% of our clothing materials are petro-based, like polyester. If we can swap some amount of those, or even industrial textiles, with these bacterially derived sheets of textile, the environmental benefits would be substantial. And frankly, when it comes to textiles, I have no particular attachment to polyester. We've had synthetic fibers like that for just less than 100 years. I'm fine swapping it out for bacterially derived materials. It just comes down to whether it's good at its job. When we talked about cellulose leather, it was all about how good it was at doing the kinds of jobs we expect from leather. Are my shoes going to fall apart when they get wet? Are they going to wear down super easily? And when it comes to clothing textiles, I will swap my polyester for cellulose clothing if it's good at, being clothing. Is it breathable? Soft? Importantly, is it pretty? Because aesthetics matter, and that's what this week's science is all about. Taking bacterial cellulose and making it pretty. So these textiles end up actually being something people want to wear. When we learned about bacterially produced self-dyeing leather, the idea was to hack the cellulose-producing bacteria themselves, K. salinus, into being able to produce eumelanin, the type of melanin that produces black pigment. Getting bacteria to make melanin had been successful in other species like E. coli, so it was just a matter of getting it to work for our little kombucha bacteria. And the results were pretty convincing. They were even able to make little patterns in the cellulose leather by making the melanin synthesis sensitive to light, then shining the light on some places and not others. It's great. The only problem is, I'm not a goth. It's incredibly cool, don't get me wrong, to be able to make self-dyeing bacterial textiles like that, but if these are gonna meaningfully swap for polyester, I need colors. And we can only get such a wide variation from melanin alone. There are two kinds of melanin, black eumelanin and red pheomelanin. Together, they account for pretty much all the pigmentation we've got. Yes, some of us may have green or blue-colored eyes, but it isn't because of green or blue pigment. Rather, it's from structures within the eye causing light to be selectively absorbed and scattered. Some colors of bird feathers work this way too. It's not that peacocks have blue pigment in their feathers, but the structure of their feathers scatters light that way. So, yeah, that's not going to be how we get a range of colors. For one, structural coloration of bacterial cellulose textile sheets sounds like an implausible approach, but fortunately, bacteria can produce pigments that humans could only dream of. Take, for instance, Chromobacterium violaceum. As the chroma part of its name suggests, it's pretty colors. The purple color comes from a pigment called violacine, which seems to play some kind of protective role for the bacteria itself. Small chemical variations in the synthesis pathway give different shades, with blue violacine, purple deoxyviolacine, teal prodioxyviolacine, and green proviolacine. Some bacteria can also make carotenoids. You might be a little more familiar with those as the antioxidant pigment that gives a lot of different veggies and plants their red, orange, and yellow colors. It turns out that some bacteria, like Rhodobacter spheroides, also synthesize these red, orange, and yellow pigments. And for both the violacine and carotenoid families of pigments, bioengineers have been able to get E. coli to synthesize them. So, our researchers have one type of bacteria, the Caetelinus, that makes the bacterial cellulose. Then we have another one, the specialized E. coli, that can dye the material. Whereas the self-dyeing leather from before was all in the one species, these scientists have opted for specialization. To accomplish this, our researchers created two teams of bacteria in co-culture. You have the Caetelinus, our textile-producing bacteria, and then there are the pigment-producing bacteria, your E. coli, for instance. They're in co-culture together, so it's like a one-pot dinner. In the same culture, both making the material and coloring it. In fact, that is the main challenge for this approach, figuring out how to get the two types of bacteria to get along and not get in each other's way. But they figured it out, and the results are quite pretty, to be honest. Sure, I could use a little more green in the green, maybe refine the blue a little, but come on! Those are some pretty incredible results for a prototype bacterial co-culture where one bacterial species produces the material that the other pigments. So, are we going to have cellulose shirts on shelves next week? Not from this technique, no. This is absolutely still in the working out the kinks and maximizing efficiency stage, which includes plans to expand the range and shades of colors. We may one day see our textile manufacturer largely move to systems like these, which would be good for a lot of reasons. The environmental ones, as I said earlier, but also because biohacking our clothing options sounds incredibly, incredibly cool. So I, for one, am excited for this advancement in bio-rainbow synthesis.